Can everybody hear me okay? You can? Okay, so I'm happy to get our trainee lightning talk started. Like Sophia said, I'm Will Baker. I'm advised by Dr. Kong, who's here, as well as Dr. Luckman. And my talk today is entitled Enhancement of Mesenchymal Stem Cell Paracrine Secretion for Cerebral Ischemia Treatment. So what is cerebral ischemia and why do we care? So cerebral ischemia, or CI, is actually a common form of what you've probably heard referred to as a stroke, specifically in this case referring to when vasculature supplying the brain is occluded, restricting blood flow, contributing to a myriad of health problems, morbidity, and mortality. Now, in terms of the epidemiological statistics of CI, it's associated with an annual incidence rate of about 800,000 Americans, and it also accounts for up to $53 billion in annual healthcare costs in the United States alone. So obviously, it constitutes a significant burden on the healthcare system. And as such, there are continual ongoing efforts to devise new treatment options that can improve the patient outcomes associated with CI. And so in terms of newer treatment options, we're in particular interested in the use of mesenchymal stem cell, or MSC therapy. And in particular, we have a keen interest in the use of mesenchymal stem cell conditioned medium, or the medium in which MSCs are grown, as these cells are known to secrete a host of different beneficial growth factors, cytokines, and extracellular vesicles that contribute to endogenous tissue repair processes, including neurogenesis and angiogenesis, which are important in the context of CI. Now, because the therapeutic efficacy of this conditioned medium is contingent on the ability of these MSCs to engage in paracrine secretion, the secretion of these biomolecules, it stands to reason that maybe we could potentially improve the efficacy of the conditioned medium by improving the paracrine secretion of the MSCs themselves. And therein lies our goal for this project. Now, common methods to influence MSC paracrine secretion, there are a range of them, including using cell culture substrates that are conjugated with a number of different peptides, the addition of exogenous growth factors, among other methods. And in that vein, we've identified two proteins of interest, specifically encadherin and FGFR1, that could potentially influence MSC paracrine secretion. So both of these proteins are found on the cell surface of MSCs, and they bind to their respective ligands, encadherin binding to an encadherin on another cell, and FGFR1 binding to its ligand, FGF2. And the binding of the ligands to each of these cell surface proteins contributes to intracellular signaling pathways that in the past have been shown to improve MSC paracrine secretion. So that's promising, but what I really want to get across in this slide is how these two proteins interact. Because relatively recently, it's become increasingly clear that these two proteins engage in a bidirectional interaction whereby encadherin binding to its ligand inhibits endocytosis of FGFR1, sensitizing cells to FGF2. Similarly, binding of FGFR1 to its ligand inhibits encadherin internalization, sensitizing cells to encadherin. So if each of these proteins is individually capable of influencing paracrine secretion, perhaps we can use them in conjunction to synergistically improve MSC paracrine secretion through this positive feedback loop. And that is exactly what we set out to do. So to accomplish this, we developed cell culture substrates with and without encadherin immobilized on the surface. And we cultured these cells with and without the presence of exogenous FGF2. We cultured these cells, extracted the conditioned medium, and assessed the neurogenic secretion according to vascular endothelial growth factor, or VEGF. Now, obviously, VEGF isn't the only neurogenic protein, but it is known to affect neurogenesis in vitro and in vivo. So we're using it as a stand-in as to give an idea about the pro-neurogenic secretion from these MSCs. So we first wanted to look at FGF2 and encadherin individually. And we found that when we treated MSCs just with FGF2, we saw a significant increase in VEGF secretion relative to untreated MSCs. This wasn't really a surprise. This has been reported not with VEGF, but with some other similar growth factors. On the other hand, when it came to culturing cells on incadherin, and this is in the absence of FGF2, while we saw a slight increment in VEGF secretion, this result did not achieve statistical significance. So that was disappointing, but remember, we're interested in how incadherin and FGF2 interact with each other. So we next cultured MSCs on incadherin in the presence of FGF2 and compared this to untreated MSCs. And we saw a dramatic, nearly fourfold increase in VEGF secretion relative to the untreated control. So this would suggest that because this disparity was significantly larger than that of FGF2 and incadherin alone, they are synergizing to improve VEGF secretion. Whether that's through the positive feedback loop we've proposed, that remains to be seen. Now, because we saw an increase in VEGF, we asked, well, can our conditioned medium improve neurogenesis itself? So we next took mouse neural stem cells, so NSCs, and we cultured them in three conditions: one with basal NSC medium, one with medium from MSCs that were not treated, and one with MSCs that were treated. And we found it consistent with our expectations that the treated group exhibited faster neurogenic differentiation, as well as improved neurite growth, suggesting that the changes in paracrine secretion we saw may in fact be contributing to appreciable changes in neurogenesis. So that was all promising. Where do we go from here? Well, of course, VEGF isn't the only neurogenic protein, and neurogenesis isn't the only process that we care about in the context of cerebral ischemia. So in addition to looking at the secretion of other growth factors, cytokines, and extracellular vesicles, we also want to test the ability of our MSC conditioned medium to promote other processes that are important in the context of cerebral ischemia, such as angiogenesis for new blood vessel formation, apoptosis for neuron survival, and finally, immunomodulation for the suppression of a deleterious immune response. And with that, I thank you. I think I got it in time. I want to thank the Kong and Lechman labs, and of course, MBM, and I'm happy to take your questions. Okay.
Will the do you think the androgenic response will occur rapidly enough to be beneficial in this case? Or would this be long-term treatment before there's actually an ischemic event?
So it's not really established in terms of like clinical setting how MSC conditioned medium is actually used. It's still pretty much in its infancy. For the most part, most clinical trials are looking at MSC, like whole MSC transplantation. So I'm honestly not quite sure, but that's something we're definitely interested in looking into, and we are going to be using an angiogenesis on a chip that I believe is actually similar to one you've used or you made to look at the angiogenic response. So yeah.
Great job, thanks.
Thank you.